No logramos hacer andar esto, pero a ver. Bueno, si no anda, agarrá el mouse y le mandás a la flecha. Un poquito vamos avanzando. El último día, el miércoles, va a salir todo bueno.
No, from there. Yeah. Yeah. No, but now, and you can pass slides too.
Pablo, ¿este es tuyo? ¿Esta botella?
Esta la compartimos.
No, acá hay otro, acá hay otro.
Ya no he puesto.
Bueno, pero acá está así.
Hola.
Buenísimo.
Hola.
Empezamos.
Hola, hola.
Bien. Ok.
Hola.
¿Qué se tocó?
No.
Le dimos el micrófono.
Hola.
Sí.
Hola, ¿qué tal?
No, acá no tocamos. Lo único que estábamos con el micrófono en la mano.
Sí, no sé.
¿Me escuchan ahí?
Estos problemas técnicos. Y ahora sí, todo ya.
Estoy acá,
Sí.
yo estoy acá, sí. No sé si me escuchan.
Estoy yo acá.
¿Dónde está el otro micrófono? ¿Está por ahí abajo?
Yo estoy acá, sí.
Y a ver el tuyo.
Sí. ¿En YouTube? Sí, por lo menos.
A ver, pásame la esta solamente. Está bien, ya le.
Dale, mira, te paso.
Sí,
I interfere.
Problemas técnicos.
Ah,
¿Puedo moverlo acá?
sí.
Ahí lo llamé. Un retardo de lo que yo digo viene.
A ver, ahora.
Okay. Well, good afternoon. We are going to start with the symposium in the afternoon. And first speaker is Pablo Iribarren. He is a biochemistry and immunologist, from the University of Córdoba. He did a postdoc at NIH in Maryland, and he is chief professor from the Facultad de Bioquímica at Córdoba, and she's a cognitive researcher at the CCD. Okay, well, thank you very much. You have 30 minutes.
All right. So thank you very much to the organizers. We are most of us friends that we met Wednesdays in the GLIA club many times by Zoom, but now we had the chance to meet face to face. So let me see. Well, first, Also, I would like to thank the collaborators, especially Liliana Cancela, Soledad Selec from Córdoba, Jimmy Wan, Joe Oppenheim, and NIH. Nicolas Tistakis, that is a key collaborator in the autophagy field. And Yannick Schwab, that allows us to do some very nice live claim studies, the funding. And today, the talk, the first, I will tell you about some game that can play microglial cells with autophagy. And I will start telling you the story of these two players. Then the longer part is some results that we have related to the effects of autophagy on microglial cell activation. And finally, when the transition from the second part to the third one, you will see some conclusions, and I will say, okay, but there is some other slides, but don't be afraid. If you are bored, there will be just 3 or 4 more. So the third part is the shortest. So long time ago, especially when I was in the postdoc, many people asked why, because it was a matter and discussion at that time, why inflammation is important in many kind of diseases if when you analyze the cell injury causes, immunologic was only one of them. So we deep dive in this concept, and we saw that all these injuries in the tissue produce some primary tissue injury, and this immediately start to activate inflammation in most of the tissues. And this inflammation is important because it's one of the activators of tissue repair. And this tissue repair, if you are lucky, you finish the story and everyone is happy. But sometimes the tissue repair is not okay, and this could produce some secondary tissue damage. But in addition, inflammation could induce itself if it is persistent in the time, secondary tissue damage. So that's why inflammation, independently of the primary injury, is present in many kinds of diseases. And this is particularly important in diseases that are developed and progress for years and years. And this is very special for neurodegenerative diseases. Well, we show one of the players, microglial cells. And microglial cells normally are playing some roles in homeostasis, CNS. But this, if we start maybe here, and the person that is ill, a disease, a neurodegenerative disease like Alzheimer's or Parkinson's disease, the condition, we know that activates inflammation. When inflammation is activated, this activates microglial cell to a pathogenic phenotype, and this pathogenic phenotype contributes again to the disease. All these diseases are share some features, that is the increased amount of amyloid proteins that are present in the tissue. Yeah, there are the amount of these proteins, these abnormal proteins, are higher than normal, and these proteins contribute to all this cycle. But we can see the cycle on the other side. We study the disease in the patient of course, in this case, are the autopsy. And we can find some microglial cells that are activated. And microglial cells, when they are activated, they produce a lot of inflammation. They coordinate inflammation, and inflammation itself, as we see before, contribute to the disease. So it's a two-way cycle that, even if in these cases the inflammation is a low-degree inflammation compared to other tissues like liver or outside the CNS, but this takes place for years. So this low-degree inflammation, years and years and years and years, maybe is not the primary cause of the disease, but contributes to the progression of the disease. The other player is autophagy. I'm going to go fast with this. Autophagy, and here I'm showing the canonical autophagy, is macroautophagy. Autophagy is a process in which inside the cells they form a vesicle that is called autophagosome that uptake, for example, proteins or even vesicles or organelles, mitochondria that are in no healthy way. So this process take of this then potentially dangerous for the cells products and fuse with the lysosome and form the autolysosome, and all these molecules, cargo molecules, are degraded and recycled. So if we see in PubMed, for example, autophagy and neuron, we search and we find about 10,000 papers that started in 1976 and 7. If we change autophagy astrocytes, the story starts in 1984. So neurons always go first, then the astrocytes. And microglial cells, there is only one paper in 1986, but the real story started in the 2000. And in the 2000 was the apoptosis era. So everybody was talking about cell death and cell death types. So we defined different cell death types, and autophagy was considered a cell death type at that time, or maybe a mechanism for a cell death survival. So in 2001, just 2 months before the 9/11, I had a chance to move to, so I entered to CONICET as a researcher here and then moved to the United States. So I worked 4 years at the NIH. And at that time, I started to study microglial cells, and we described all these pathways related. Sorry, the pointer, I think I have not option here to change to the pointer, so yes, well, that's okay. So we describe in microglial cells how the activation of all these toll-like receptors activate different pathways, intercellular pathways, signaling pathways, that promote the FPR2 gene expression. And this gene code for a protein that is a 7-domain transmembrane protein. It's a chemoattractant receptor that works as a chemoattractant for amyloid beta 42. So that was the protein that deposit in the case of Alzheimer's disease patients. So we described all these things, and I work in the NCI. It's a National Cancer Institute. I think I was lucky because when I returned to Argentina in 2005, after a site visit, the lab was pressured just to move to cancer because it was a national cancer institute. So this allowed me to continue the project here with collaboration of my former boss. Of course, we signed many agreements and restrictions in some ways, but we continue to produce some studies here. And the first we asked was, can folate receptors control microglial cell population by affecting these cells' viability? So we saw in the cell cultures when we stimulate, for example, in this case with toll-like receptor 2, that this cell produces a lot of pro-inflammatory molecules. But later in the cell culture, they die, and the aspect of the cells are really, really weird. And remember, this was in the 2000s, so everyone was studying cell death types. So we believe that maybe autophagy was one of the causes, because when we see on the bottom, when you can read PGN, this is annexin 5 PI, well, 7AAD, similar to PI. When you see the classical apoptosis in the cells, in the kinetic of the cell death, you have a time point where the cells are single annexin 5 positive cells. But it wasn't the case in our culture where we stimulate with PGN. They go from living cells directly to double positive cells. So it looks like necrotic cells. So we use LC3 marker. That is a classical marker to identify autophagy. You see in the Western blot, the second band is named LC3-2. There is the version of LC3 that is located in the autophagous. So we found that 48 hours after stimulation, the LC3-2 band appears, and the specific autophagy inhibitor blocks that pattern. In addition, when we added the inhibitor to the cell death, so the cell survived. So we suspect finally that autophagy was playing a role. And then we, you see on the top, when we stimulate with PGM, the LC3 pattern of staining by confocal microscopy moved from diffuse cytosolic staining to what is called punta. It's spots like very positive for LC3. And of course, we wanted to check that was a toll-like receptor 2 specific. So we ran the experiment with toll-like receptor 2 KO primary microglial cells, and in these cells, PGM failed to induce autophagy. But in the autophagy field, we cannot just measure with LC3 and LC3 positive and say it's autophagy. We need to demonstrate autophagy flux. One way to demonstrate autophagy flux, the autophagy flux is to, for example, demonstrate the colocalization of LC3 with the lysosome marker. In this case is LAMP1. And you see nice on the bottom, the nice colocalization. So this indicates autophagy flux was taking place. Then all this was made in vitro. So then we asked whether if we inject in the CNS environment, the story should be the same. So we inject PGN or PBS as control at different times, took out the cells for flow cytometry, and also we made some slices to do some confocal. And we saw that a year of the injection, in the ipsilateral site of injection, the appearance of LC3 punta, similar than the previous studies in vitro, and colocalized with the lysosome. So autophagy flux was taking place in the brain, and we didn't know which cell type. So using flow cytometry, we could measure the cell death obtained from these brains and stain for markers of microglia. So we gave microglia cell, and we demonstrate that at 48 and 72 hours after injection, microglial cells were dying. And also, if you see in the bottom, the tissue, we stain with CD45, that is only positive in the parenchyma for microglial cells. These spots where the density of microglia is lower than the control. So inflammation affects the uptake and degradation of aggregated proteins, and this could be a very interesting topic for neurodegenerative diseases. Compromise major signaling pathways like MAP-kinase or NF-kappa B signaling pathways, and also could affect inflammation, cell proliferation, and cell death and survival, as we saw before. So we, at this point, we asked whether autophagy can modulate that. It is another different question. At that time, we were thinking, okay, we can modulate, we can exploit autophagy to modulate neuroinflammation. So we induced autophagy with rapamycin, and we saw the punta pattern after rapamycin stimulation. Is a classical mTOR target that activates autophagy. Here you see the 3D that in the control we didn't see colocalization, but you see some nice colocalization, and the autophagy flux was taking place. So this was just to be sure that we could induce autophagy, and also we induce autophagy with triallose. The triallose is an mTOR independent way to activate autophagy. So we measure and we obtain the autophagy pattern in the Western blot, and the colocalization indicated the autophagy flux. Then when we have the microglial cells that we increase the level of autophagy in these cells with these pharmacological stimulations, we check whether these cells can produce pro-inflammatory molecules. When we, in these cases, sorry, no pointer. The tall bars indicate the stimulation with alpha-synuclein, that is an amyloid protein that is typical of Parkinson's disease in substantia nigra. And alpha-synuclein induce very nicely IL-1, IL-6, nitric oxide, and TNF-alpha. But if the cells, before the stimulation with alpha-synuclein, was higher rate of autophagy induced by the pharmacological stimulation, the inflammatory response didn't take place. So then with these cells, we treat first the cells, and then we added some neuronal origin cells and check the viability of both cells, of microglia and neurons. And we saw that in the presence of microglial cells, neurons start to die. But if the microglial cells that we added had a high rate of autophagy, these cells couldn't kill the neurons. So And this process also was dependent of p38 MAP kinase inhibition. That is the last two bars. So indicating the MAP kinase was part of this pathway that was involved in the neurotoxicity. And then we made focus on nitric oxide, and we found that if we added nitric oxide inhibition, they block the neurotoxicity. So the autophagy block the nitric oxide production, block the iNOS gene expression, and prevent the neurotoxicity. So alpha-synuclein induced MAP kinase activation and pro-inflammatory mediator. So I'm doing talking about this story for short, but it's a longer story, no? So alpha-synuclein induced MAP kinase and pro-inflammatory mediator with some neurotoxic effects. So we at that time we were thinking, okay, if alpha-synuclein goes and is endocytosed by microglial cells, it could be a chance that microglial cells can degrade alpha-synuclein inside. So, but what happened with autophagy? And we saw at the time that autophagy could be a good mechanism to clear the amyloid inside the cell. So first we wanted to know if alpha-synuclein could induce autophagy. And we have a lot of filmation, deep microscopy, and in minutes after we added alpha-synuclein to the microglial cells, goes directly to the lysosome, for our surprise. And 12 hours after that, autophagy, that is showing here, and here, this is the localization of alpha-synuclein, LAM, it's a lysosomal marker, and LC3 molecule. So all this takes place 12 hours later that we saw the alpha-synuclein inside the lysosome. So this was strange for us, and at that time, the autophagy people asked whether, okay, is this really autophagy? Because it could be a lab. Lab is a LC3 associated phagocytosis. Okay, but we said, but this LC3 can later. We film the alpha-synuclein and first goes to the lysosome. But anyway, a nice and elegant way to demonstrate that is to do a clem, a limb clem. So we have the chance, and we were lucky to be one of the first to show that alpha-synuclein. On the left, you see a giant lysosome with a single membrane. With this technique, you can correlate the transmission electronic microscopy with the immunostaining. So in red, you see alpha-synuclein, and in green, you see LC3. At this point, the image that is green is this little square with red and green is the same that you see in the big square. So you see the LC3, a nice double membrane structure, is autophagosome, very clear. And at that time, they are separated. So you have the autophagosome and the lysosome. They are not fused. So then in the other one, these are microglia, of course, stimulated with alpha-synuclein. You see the nice colocalization in these structures. You see multilamellar layers of membranes. So that indicates is typical macroautophagy. And see you see the tomogram of about 20-25 slices. You see the autophagosome containing a lot of vesicles inside. And also we could detect that this autophagosome, you see the multilamellar layers forming. And the tomogram shows that the ER membranes are very close to the autophagosome, indicating that the ER is the contributor of membranes. So these structures need membrane donors. And this, in this case, for us, it was the ER. So later we demonstrate, and here, so it's difficult, sorry, without pointer to describe that, but here we demonstrated that alpha-synuclein induced lysosomal damage with the galectin-3 spot-like pattern and colocalized with LAM1, and also colocalized with alpha-synuclein and LC3. So this marker of lysosomal damage, and we knocked down autophagy with siRNA for FIP200 and a pharmacologic inhibitor of autophagy, and we measured the mitochondrial health. So when we took out autophagy in this microglia stimulated with alpha-synuclein, the mitochondrial health was impaired. So in addition, we start to detecting some apoptosis of microglial cells, and also you see downregulation of BCL2 and BCL-XL that are protective factors for apoptosis, and increase caspase 3. So we propose that alpha-synuclein enters, it goes to the lysosome, and later this lysosome that cannot degrade and eliminate this alpha-synuclein start to be damaged, and this damage is very dangerous for the cell. So these damaged lysosomes activate autophagy, and this autophagy protects the cells of cell death. But in additional result that I don't have time to show, this alpha-synuclein couldn't be degraded in the cells even if you stay longer. But the cells survive, and the alpha-synuclein is still there. And we have some movies showing that these cells are transmitting alpha-synuclein to other neighbor cells. Yes, just few more. So autophagy, this is just going back to all the studies. I will say something short. Autophagy affect many different things related to microglial cell activation and neuroinflammation. And why is that? So the reductionist way to think is, okay, autophagy do this to inflammation and that's it. No, because autophagy is a fundamental process in the cells, affects all types of cells, and the effects of autophagy depends of the cell type, depends of the time, depends of the pathway, depends of the amount of stimuli, and so on, and so on, and so on. So that's why we detect many different things on microglial activation, but the important thing is that this open maybe the chance for targeting autophagy in microglial cells for possible treatments. So just few slides and finish. This is, I will take the liberty to share with you some concepts. So there's a study, a very nice study. We are living in a world that we are increasing the computing capabilities. So we are generating data. Here, everybody of us are generating data right now. If you go to the mountain and they say, okay, I want to disconnect. So do I generate data? Yes, you still are generating data. If you see the captcha that choose traffic light images, and you need to click and ask you, are you a robot? So we believe that they are scared if we are a robot. But no, when you pick all these traffic light or bicycles, for example, you are training models. You are training models that could be used, for example, for autonomous driving cars. We didn't know that. But the way that we move the mouse and click and select is different that a machine learning model could do that. So for free, we are offering this service of these companies and training models. This is our era, and research have a lot of data and resources that are unused. So the calculation of these nice papers in the Cell publication environment of 2 years ago say that 6.2 billion of resources in science are unused. And many of these are data. So, sorry, it's 2 more. So NIH started 2 initiatives. One is a grant for potential data reuse, and the another one start seminar series. When all of NIH start seminar series, it's because they are interested in this field. So this and one more and finish. So Marco Prince, 2 years ago or 3 years ago, I think in 2019, wrote and published this very nice review about microglial cells, and he finished saying that, in particular, for question for the field, open question for the field, the discovery of context-specific microglia transcriptional states is exciting, and we provide better understanding of CNS diseases. He paid very much attention to this point. Here, you see, this is Clarissa, a PhD student. You see a classical volcano plot or lollipop plots that you see differentially expressed genes. I mark TNF and INOS, for example. And you can associate this with gene ontology and associate the expression of the genes with pathways or cellular responses. Also, you can use the KEG protocol. The KEG is a very nice initiative in Japan, KEG University, that take and provide you with all these pathways. And in these pathways, you run your data set and see whether are in green, the gene is down-regulated, in red are up-regulated. And you see when you stimulate microglial cells in the knockout mice, these are pro-inflammatory mediators, are mostly down-regulated because they have no proline receptor. But the wild type, all these pro-inflammatory markers are red. So this is just transcriptomic, this RNA-seq. But you can do proteomics. You can do many other studies of the omics area, and this is bioinformatics. But what do you think if I say that, okay, in this kind of Venn diagram, I finish. Venn diagram, where are we? Where are we in domain expertise? And we have the data. If we see, okay, my lab collaborates with the bioinformatics lab to help me to do these studies. But what you say if I ask it, okay, can you add to this analysis health records of the patients? Can you analyze all together? Can you integrate all this information? This is the big challenge that we have. And if we, as a scientist community, don't take this place will be taken by the companies. So I just suggest, sorry for suggest this, but that everyone should be involved in learn how to analyze data, because this is what is coming. In 5 years, I put this to relieve, but in 5 years, we have the promise of have quantum processors that can have the speed that maybe in seconds analyze all this big data that we take sometimes days. So this is a challenge. So I wanted to share with you. Yeah, I'm sorry for making this too long. And many of the information I did for short because are complicated experiments. So I invite you later, maybe, if you are interested, to discuss with me. Okay, and share and talk about this. Thank you very much.
Okay. Only one short question for Pablo, please.
Pablo, you have shown that alpha-synuclein is not well digested by lysosomes, so they have the model cells. Do you know if inflammation has any implication on that, or it's just the conformation of alpha-synuclein that makes the whole process complicated?
No, but it's a very nice question. So the shape of alpha-synuclein is very important to form. So we check monomers and we check fibrils. So monomers did not affect microglial cells, but fibril induce pro-inflammatory molecules and go inside the cell and stay there and keep the cell alive. It's like a parasite that is keeping the cell alive. Maybe if we think in this prion theory that maybe keep the cell alive to keep transmitting alpha-synuclein to neighbor cells, maybe. But this was the situation. I don't know if you asked me something else. Yes, that was fibrils.
I have a short question, but I didn't take that. Everybody.
The problem is that I received 15 emails asking me how many time I have. I said 30 minutes for 30 minutes and 5 minutes for question. And nobody is in the schedule. So
okay, don't worry. I tell you it was okay. I leave it. I tell you. No, I just wanted to comment that when you activate the mitochondrial fragmented in the microglia, this change in the mitochondria morphology may have something to do with the other part also. That was my question, but you can answer later. Okay. So let's move to the next speaker. And so finally, well, next speaker is Guillermo Giambartolomei. He is doctor in immunology from the University of Buenos Aires, and he did a postdoc at Tulane, USA. And now he is a main researcher at the CONICET, and she's chief of the immunopathogeny lab at the INCHE. So thank you for coming. And you have 30 minutes, please.
Yeah, I know. I know. I have two colloquial things to say. First of all, Felipe Curtis here, so as Ruth has said, put the water in the pasta because the house is full. Second, I want to thank the organizers, especially Janina, for his stamina and his energy driving all us here. And as we said, she is the founder of immunochemistry. I will name him Our Lady of the GLIA from now on. I am agnostic for the one that are not. She will be named as Our Lady of the GLIA. So I'm waiting.
Sorry, in America, she said,
okay. I'm sorry, I didn't know. So the only thing I see here is my picture of myself. Okay. So I'm going to talk about, you will have a taste of immunology after Paulo and me, and I think Hugo will maybe have something to do with that. I'm going to talk about microglial phagocytosis as a consequence of, or as a cause of neurodegeneration or neuronal death. Just to let you know, let me see in here. Okay. You know that microglia are the residence macrophages of the CNS. So, and they're specialized in phagocytosis. So they eat things, okay? They can eat on phagocytose excess of synapses and trim and prune. There's no pointer here. I'm sorry, I see a pointer, I don't see it. During development, but also they can protect the central nervous system by eating pathogens like viruses and bacteria and dead cells and debris and amyloids, etc. However, excessive activation of microglia can drive the phagocytosis of, for instance, myelin, as is depicted over there, and even the cell body of a neuron, causing neurophagy. And this is what I'm going to talk about today. Yes. Oh, yeah. Thank you very much. I was thinking about that. Yeah. You can see this in here. You can see. Okay. Let's see. No. Okay. Okay. Yeah. No. But I want to. This is not the problem. Thank you very much. Okay. A lot of years ago, she know why analysis studies have a link microglial phagocytosis with Alzheimer's disease. That means that a lot of pathways related to phagocytosis were genetically linked to Alzheimer's disease. Of course, it doesn't tell us if phagocytosis is good or bad or both. Maybe we know nowadays that it's both. As Dr. Sun has said, microglia have many different states, and they are not either bad or good, as we have things for many years. Another important issue is how the field have became interested in this. Why it's not working? Okay, there is that in many neurodegenerative diseases, particularly in Alzheimer's disease, there are a lot of loss of neuron, brain atrophy. Okay, and brain atrophy, of course, without significant neuronal death. By what I mean is no more necroptosis or apoptosis or necrosis of neurons. That drives, and there is little evidence that preventing neuronal death by blocking apoptosis, for instance, prevents this neurodegeneration, this brain atrophy, which leads us to the concept that maybe it could be microglial phagocytosis of leaked by stress neurons, what is driving this brain atrophy and the losing of neurons. And this is going to what I'm talking to today, okay? How our lab get involved in this? Okay, we work in a disease, in a bacterial disease called brucellosis that has focal manifestations. And when this focal manifestation affects the brain, it's called neurobrucellosis. And what happened in neurobrucellosis is that most of the symptoms that patients suffer are related to neuronal loss. More interestingly, more recently, stranded dolphins, that is the only natural species that also with humans suffer neurobrucellosis, were reported to have neuronal damage. So that there should be a link between microglial activation by Brucella abortus and the loss of neuron, okay? And we were investigating this for many years, and we have discovered by co-culture in different glial cells with neurons that microglia were phagocytosing neurons. This is no surprise. What was surprising is that these neurons were alive, or at least they don't show any signs of apoptosis or necrosis. It took us a while to figure out the mechanism by which this microglia, and I'm going to talk about this because I talked about this the last GLIA meeting, and Janine is going to kill me if I spend about 30 minutes, but it's easy to see in this scheme, and what we found out is for this microglia activated by the infection to kill neurons, it takes two things that are very important. The activation of this microglia leads to an augmentation in phagocytic capacity, and two, the release of pro-inflammatory mediators. More important, it's not working, the production of nitric oxide that stresses the neuron, okay, pop up in the membrane for fatty glycerin that is recognized by the opsonin that uses the butyrate receptor to digest the neuron. So let me show you just two simple experiments that show that these two events, nitric oxide production, we are not prepared this talk with Pablo. Pablo has talked about nitric oxide a minute ago, and the increase in phagocytosis are important. First of all, if you culture microglia and infect them with Brucella abortus, neurons are much more, it's not working. The red column shows that they are dead, while if you don't infect the culture. But if in the media you put inhibitors of, nitric oxide production, like aminoguanidine or an ester called N-amine, the death of neuron is gone. On the other hand, if we block the butyrate receptor capacity of bind through this opsonin to the neurons by using a circular 3 peptide that's called CRCD, the death of neuron is gone. What is important, I want to point out in this picture, is when you block phagocytosis or the production of nitric oxide, the remaining neurons are not either apoptotic or necrotic. They are alive, indicating that phagocytosis is the cause and not the consequence of this. An important question we have is, it is special of Brucella abortus? The answer is no. Okay, if we treat microglia with LPS or lipoproteins, TR4 and TR2 ligands, or we infected them with a virus, Zika virus, the microglia is able to phagocytose neurons and kill them. And there should be in the culture microglia, because if you don't have microglia, the virus by itself do nothing to you. They also happen with another bacteria, and also Guy Brown has shown for many works that beta amyloids, oligomers, and alpha synuclein and other stimulators of microglia that are related to neurodegenerative diseases can do the job. Okay, is this primary phagocytosis? The answer is yes. If we block it with aminoguanidine or with an exin-5 that blocks PS, the death is gone. So for many years, we wanted to know how this mechanism was controlled. We are immunologists. We say, okay, this should be controlled by some kind of cytokine or a soluble factor. And we finally found out this by the indirect way, by indirect means, as usual. because we are interested in the relationship that astrocytes and microglia might have in our infection. We have listened to Shane Liden's talk last meeting in which he showed us that activated microglia with LPS was able to induce a state of inflammatory state by which many mediators activate these astrocytes and render them able to kill neurons by phagocytosing them, okay? And we want to see the opposite way, because we know that also that our bacteria infect the astrocytes. So we say, okay, in which way can we prove that astrocytes are doing something to microglia? And we do that this in vitro by a simple experiment in which we infect the astrocytes with our bacteria, with Brucella, take the supernatants and simulate co-cultures of microglia and neurons and see what happens a time later. And what we see is that supernatants from astrocytes infected were able to kill neurons if they co-culture with microglia in a time and dose-dependent manner. And this doesn't depend on a special culture, primary culture of microglia, astrocytes, or neuron, because 5 different cultures from that different animal, no matter the sex of the animal, no matter the timing of whatever, the phenomenon was taking place. What is more important, is that for death to occur, microglia should be in the culture, because supernatant by themselves do nothing. Okay? So the next question was, what are these supernatants doing to microglia? And what they were doing is when they are activating them, inflammatory activating the microglia. And you can see with you by qPCR and ELISA, you can see that the microglia culture with the supernatant of the infected astrocyte increases the secretion of pro-inflammatory mediators and, more important, nitric oxide. Okay? And also, the phagocytic capacity of the microglia was also increased when we treat them with supernatants of the abortus infected astrocytes. So the following question was obvious. This is also primary phagocytosis. Is this astrocyte inducing the same phenomenon we have seen when we infect the microglia? To make a long story short, the answer is yes, because when we treat them with supernatants, but in the medium we put an X5 that blocks PS, we block the interaction of the mitonectin receptor, and we block the simulation of the nitric oxide synthase, then that is gone. Okay? So, by all means, this experiment are telling us that there is a solvent factor in the supernatants that is inducing this. So we wanted to figure out which one, and we did a simple experiment using neutralizing antibody, because we know that astrocytes secrete many inflammatory mediators among them. And for our surprise, we have previous results indicating that TNF-alpha and IL-1 beta have something to do with the activation of microglia and has impact in the brain barrier and also in astrocytes itself, but we have never paid attention to IL-6. And to our surprise, when we incubate these co-cultures activated as supernatant with an anti-inflammatory, with an anti-IL-6 neutralizing antibody, the neuronal death is gone. So Julia, during pandemic, was our student that do the experiment, went to a meeting, and someone raised a question. Said, I have a question. How do you know that IL-6 is coming from the supernatants and not is coming from the microglia itself? Because you show that microglia, when you infect it, also produces IL-6. And the design of this experiment with using a neutralizing antibody has no clue of a way of resolving this question. So we hate that question, but it pushed us to another situation that was really interesting. And with the help of Juliana Casatar and Oscar Campetela, we were able to find out knockout IL-6 knockout mice that are really, really hard to breed, really hard to breed. So we pray to God and to our creator or whatever, and we find many pups, and we can find microglia and astrocytes and culture them, and we do the simple experiment. We cross the wild-type microglia and treat it with the knockout IL-6 knockout supernatant and do the opposite experiment, see what's going on. And I'm challenging you to see what was the answer of this experiment. Is the microglia guilty or is the astrocytes guilty? And you know what? There are none of them, there are both of them. Because when we use, supernatant from wild type, again, you see neuronal death. But if supernatants come from IL-6 knockout mice, although the amount of death is still significant from the non-infected ones, is less and significantly less than the wild type, indicating that something is going on also, and what's going on also is when we do the other experiment using a microglia that is IL-6 knockout, it's happening the same thing. More importantly, if we knock out both, the death is gone. We say, okay, is this by standard activation telling that IL-6 is important? So it is important also when we infect directly the microglia, and the answer is yes. We infect microglia, and we infect knockout microglia, and the death is gone. And also we neutralize it with the antibody. So I was telling you that there is two things necessary for this neuronal death to take place. One is phagocytosis. The other one is oxide production. The following question is, okay, in which of these two IL-6 is involved, if not in both? Okay? So for this, we perform very classical and simple experiment, and we find out that IL-6 was involved in neutralizing the phagocytic capacity of microglia and not the production of nitric oxide. Okay? Indeed, if we treat, and there's no infection here, and we treat microglia with IL-6, recombinant IL-6, as you can see, that is an increase in the phagocytic capacity, but not in the production of nitric oxide. And also, there is no capacity of these microglia to kill neurons, because for more that they are more phagocytic, they cannot release any inflammatory activation, there is no nitric oxide, and the death is not taking place, indicating that IL-6 is necessary but not sufficient to induce this primary phagocytosis. So for any immunologist in the room, not Pablo and not my students, the following question is, in which way IL-6 is doing this? Because if you don't know, I will tell you that IL-6 has two ways. It has three, but two classical ways of inducing activation. One is called the classical activation, because IL-6 receptor doesn't have an intrasignal transducer. So it has to use another molecule called GP130 to transduce signal. And when the IL-6 molecule binds to this complex, it's called classical. Yet, IL-6 receptor can be released to the median and form a complex with IL-6, signaling through a GP130 molecule that is ubiquitously expressed in many, many cells. So the only way to find out this, because neutralizing antibodies block both signaling pathway, is using a chimera of an Fc receptor together with a molecule GP130 that specifically blocks the trans-signaling. And we did that, and of course, or luckily for us, why not working? Oh, here. The GP chimera is blocking as well as the neutralizing, indicating that the way in which IL-6 is doing this is by trans-signaling. Okay? So, is it blocking also phagocytosis? The answer is yes. The GP130 chimera blocks the phagocytic capacity of microglia when it's activated in the standard way, but also when we activate it by infecting the microglia with our bacteria, indicating that IL-6 trans-signaling is operating in only the augmentation in the phagocytic capacity. So the following an obvious question is, I knock out the lab and say, okay, who is regulating nitric oxide release. Could be IL-6, well, it couldn't because we have shown in the last picture that it was not involved. So to make a very, very long story short, we have a result many years ago of this, in which we could block the primary phagocytosis by blocking the type 1 interferon signal. Okay, this is interesting. So could it be also when we do it in the bystander way? The answer is yes. So it seems that the interferon signal in bad way is very important in this. And so let's do a classical experiment. Could be this the one that is involving nitric oxide releasing? The answer is yes, as you can see here. When we block with an antibody the receptor of the type 1 interferons, the capacity of the microglia infected or treated with an supernatant astrocyte is gone, while its phagocytic capacity remains untouched. Okay, so what we have up to here is this. We have a black box. We know that interferons bind to the receptor, and on the other side of the cell, there is a release of nitric oxide. So what happened in the middle? In the middle, most probably, there was a pathway that transformed this signal into the activation of the inosine. We proved that this to be true, and also the production of the protein. So interferon is inducing the gene of nitric oxide synthase, inducible nitric oxide synthase. This is particularly expressed in microglia, and inducing the production of the protein to induce the release of nitric oxide. So, in which way? It could be many proteins in there, and I will talk about one in here and invite you to see the poster of Julia Rodriguez after our talk, because I won't spoil her results. So I give you a little bit of the taste of what's going on, because we have many transcription factors and kinases involved, but when you go to the literature and look for interferon gamma, type 1 interferon receptor, the molecules that pop up are STATs. Okay, I'm sorry, next one. Are STAT1 and STAT2, and the complex that makes the transcription factor for the inducible genes of interferon. And we see what's going on by Western blot, and as you see in here, blocking the IFNAR receptor also blocks not only the phosphorylation of STAT1, but also the induction of the protein. So I want to end here, okay, for the one that are interested in our job, in our work. There will be a poster indicating that many other factors are involved as kappa beta, and also many MAP kinases in this story of the interferon and the inosine. So to wrap up, we can say that at least when inflammatory activated by the bacteria, in this case, microglia come in the surroundings of the brain parenchyma being activated directly by an agnoxious stimulus, but also by standard, but other cells that are also activated. You know that of immunology, and what is important that we have dissected two cytokines that control independently in a single cell two different mechanisms that are important to a very important phenomenon to occur, that is the death of a neuron. Okay. I already mentioned Julia Rodriguez, our student. Also Ana Rodriguez, that has discovered this phenomenon in our lab. Other people in the lab, our collaboration, I will thank to our collaboration, Sergio Porta Ribeira, and by the Dennis in the States. I have already mentioned Juliana Oscar that gave us the IL-6 mouse, all the financial support. And I will thank you very much for your attention. I will take any question you may have. Thank you very much. Okay.
Yeah, thank you. It was less than 30 minutes. This is great. So we have time for 2 questions then. We have questions for Guillermo.
Thank you, Isha. Very nice talk. Neurons are very large. So where does it start? Is it the soma that gets eaten first?
Okay. Very good question. Everybody, once in a while, this talk, same question arises. First of all, neurons are large, but microglia can increase up to 20 to 30 times its volume when it is activated. We think it starts with the axon and the dendrites because it's where we see the most expression of PS, of acetylserine, when it's harmed by the activating microglia. We don't see many PS at the soma, and it goes along with the trimming and the dendrites. But what we see inside the microglia by confocal microscopy is a chunk of a neuron. I don't know, it's the accumulation of dendrites and the neurons are gone of the culture. Okay, I don't know if you answer your question. Thank you. Yeah. Fernando.
Thank you, Guille. Very nice talk. I like it very much. I like also the rowing figure there.
I don't have dulce de leche. I don't have dulce. I have to add dulce de leche at the bottom.
Yeah.
It's toffee because it's thicker. We are, you know, it's not against the string. We are rowing in toffee.
Maybe I missed the link. Can you maybe wrap up and make a link between interferon inducing NO and the IL-6?
Yes. For this to take place, for the mechanism, it has to happen 2 things at the same time, concomitantly. An increase in the phagocytic capacity together with the capacity of the same cell to release at least NO. Because when you activate it, when you inflammatory activate the microglia, it happens many things. Release of inflammatory activation, NO, plus the inflammatory, plus the phagocytic capacity. We didn't do the experiment, it was already done, but if you take it microglia and independently of the neuron, you activate it, okay, you augment the capacity and the production of the NO. But you take it and put it later in the culture of healthy neurons, NO is a gas, it's not there anymore. So this activating microglia with its capacity of increased capacity of phagocytosis is enabled because the neuron doesn't expose emitting signals. So the NO, what we have figured it out is not only the mechanism of how it's regulated. We have figured out pretty much how it's regulated the production of NO. We don't know what's going on with IL-6 phagocytosis because phagocytosis is a much more complex mechanism. And we want to do bulk RNA and other stuff because we think that beyond Brucella, it could be involved in neurodegeneration diseases. There is a work, there is many receptors, not only mitotic receptor of Guy Brown showing that in vivo in mice, in a model of alpha-synuclein, by blocking one of the receptors, the symptoms are gone and the length of neuron is recovered. So, I don't know if you answer your question. 2 things concomitantly has to happen. An inflammatory activation and the release of nitric oxide, or another sialic acid is also a don't-eat-me signal. Activating microglia release neuraminidase that desialylate this and make it eatable. Okay? So inflammatory activation release things that expose eat-me signals in neurons. Plus this activation goes along with the augmentation of the phagocytic capacity. We have learned in our lab that these 2 phenomena are regulated by 2 different cytokines.
That makes a lot of sense. We've been talking about the 2-peak model for Parkinson's for a long time, but if you, well, not we, everybody.
Yeah, I know. No, but I'm not we. we, right? No, not he.
But but if the two signals are simultaneously expressed,
Yeah.
will you then start a feedback loop that will perpetuate with time?
This is a very good question. Yeah. Yes, to a point, because this is a good thing to have good students. Julia has just, there is a woman in Italy called Francesca Petri that works with several fish. And she figured it out that when a microglia eat a neuron, apoptotic or stress or whatever, do it once at a time. And to a certain point, if you see, we have pushed the system and we only have 50% of neuron death. I don't know what's going on in vivo. I mean, people with brucellosis don't have a huge brain atrophy. Even people with Alzheimer's disease is not overwhelmed. It's just in some places, okay? And it seems that microglia has a capacity that is regulated by the central zone Francesca Petri has discovered using several fish model, and a new structure called gastrosome. So you can go from a neuron that is huge, as Lorena said, once at a time. Digest it and then go the other, and then go the other, and there's picture and films. So it will be perpetrated, but as in any single immune response, inflammation tends to be reverted by we don't have the answer but I'm sure there is anti-inflammatory cytokines and disease to control this in any way or another. And the environment will control it in a way. Okay, it will damage, but not forever.
One quick question. I'm very interested about the interaction with astrocytes.
Yep.
And you said that when you have this infection, you have reactive astrocytes also, because they also express ions and they produce lymphocytes.
Yes.
So the other way, like you said, for the interleukin-6, perhaps.
Yes.
That was my question.
Yeah, it could. Yeah, for sure, it could be. Yes,
Okay.
You're right.
Thank you very much.
No, you're welcome. Thank you, guys.
Okay.
In the meantime, if somebody wants to ask another question to me.
Pablo.
No.
Yeah, I have a question for you.
Okay, go ahead.
Yeah, because
Hugo is connected.
he's still rowing. You have seen. I get interested. I saw your talk many times. I never pay attention to. You see, ER structures comes to the autophagosome. You see ER, reticulum in the plasma reticulum. Have you looked or have you any idea of if the unfolding protein response is also triggering these cells?
You mean unfolded from inside the cell, not?
Yes.
In our response, we never check that, and we have no evidence. But autophagy have many kinds of autophagy. So you have agrophagy. This involves adapters, other molecules. In our case, because I didn't have enough time, we saw some that this effect of alpha-synuclein was dependent of EBK1 of the neuron pathway that is related to selective autophagy. But maybe EBK1 could be more in this process that you mentioned.
Thank you very much. Should I close this?
Yeah. We are going to pretend
I don't want to touch anything. Hugo is here. Hello, Hugo.
Hello.
But I see you don't see it.
I don't know to, Okay. But perhaps I can go with the presentation.
Wait. Okay.
There he is. Hello, Hugo. Great pleasure to see you. Can you hear me?
Yeah. Can you hear me?
¿Me escuchas?
Sí, perfecto.
Well, I'm going to start with the presentation of the next speaker. It's a great pleasure to me to present Hugo Peluffo. It's not because the other one didn't want a pleasure, but I work side by side with Hugo for many years, so it's nice to meet him in this way. So I will tell you that Hugo has his degree from the University of de la República in Montevideo, Uruguay. He did his PhD at Barcelona, the University of Barcelona. Then he came back to Uruguay and worked in the Instituto Pasteur and the Facultad de Medicina for many years. And now, unfortunately, he's back to Barcelona. And there, I don't know your position there, sorry, but he's back working there. So thank you, Hugo, for coming, and we are going to listen your presentation.
Okay, thank you.
Can you turn on your volume, please, a little bit?
My volume, like this. Do you hear me there? If I speak like that? Hello? Do you hear me there? Is it okay if I speak like this or?
No te escuchamos.
¿No? No sé, voy hablando a ver hasta que pase algo. Hola, probando. Hola. ¿Me escuchan? Hola.
Ahora sí.
Ahora sí. Okay. Now it's okay now? Yeah. Patricia, is okay now? You all hear me?
Sí, ahora está bien.
Okay. Great. It's great to be with you at least by Zoom. It would have been great to be with you there, but we all do what we can. So here we are. And thank you for the introduction. So, okay. Any problem? Everything okay or?
Ahora, decirle que está ok. Decirle que...
¿Está todo bien? Ahora sí.
Now it's okay?
Now it's okay.
After minutes.
You hear me now correctly? Yeah? I'm not sure if you hear me. Hola.
También, sí.
¿Escuchan bien entonces? So, everything okay? I'm sorry, but I'm not sure if you hear me correctly.
It's okay. It's okay.
Okay. So, as I was telling you, it would have been great to be there with all of you, but we do what we can, so here we are. I'm going to show you today results from our group and many collaborations we have related to C200F lipid binding immune receptor, mainly in aging, and also some new data we have related to Alzheimer's disease. So you all know well that microglia have many functions. You have been discussing this with Pablo and Guillermo, and you all know this. And we are particularly interested in the molecules that are on the surface of microglia that help these microglia to sense the milieu, let's say. Everything is okay? Should I go or? Is everything okay? Oh, okay, I wait.
A ver, prueba ahora.
Hola, hola. Do you
Ok.
hear me now? Everything okay?
Sí, igual que hable más lejos del micrófono.
¿Más lejos?
Ahí
Sí,
está.
por favor.
Un poquito más lejos.
Sí.
Vale, ¿así? ¿Así va bien? Ok. Should I continue or?
No, este no está conectado. No, está conectado. Zoom. Hola.
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 Hola. ¿Me escuchan ahí?
A ver, empiece.
Okay. So we'll try again.
No, wait.
Acá está.
Hola.
Vamos a probar a ver. Con auriculares.
Ahí está. ¿Cómo que pasó?
Pruebo con auriculares.
Se ponga auriculares
Que
porque
se ponga
aparte saliendo
auriculares.
el sonido y
Ah.
escuchamos lo que toma.
A ver, ahí cómo está. ¿Está mejor ahí? Ahora estoy con auriculares. Hola. Hola, hola. ¿Me escucha mejor ahí? Bueno, cuando ustedes me van diciendo cómo está la cosa. Okay, so let's begin again. So as I was telling you, we are interested in really understanding how microglia senses what's going on in the neural parenchyma and which molecules in the surface of this microglia coordinate the response against different stimuli. For example, like extracellular pathogens. Okay, no, no wait, we'll wait. Si apago ahora, si apago así mi, si apago mi audio, ¿se escucha mejor ahora? No sé si ahora se eco o no. Me sugirieron recién apagar los parlantes y los apagué ahora, pero no los escucho a ustedes porque como apagué el sonido, salgo. Hola, ¿se escucha ahora?
Ahora.
Hola Hugo, ¿cómo estás? Por favor, ¿puedes compartir tu pantalla?
Sí. Ahora no me deja compartir, se ve que me tienen que dar permiso.
Ahora lo deja. Te pruebe ahora.
Ahora sí.
Vale.
Ahí se ve.
Bueno, ahora empezá.
Pruebo ahora. So will you tell me we can try again?
Sí, sale 10.
Okay, let's try again then.
Sale bien, empezá.
Right, okay. So, well, as I was telling you, I'm not sure if you heard anything, so it's just really a pleasure to be here, not with you, but more or less with you. I would love to be there, but we do what we can. So, I'm presenting today some data related to CD200F lipid binding immune receptor, mainly in aging, but also in Alzheimer's disease, as we have some new data that I wanted to share with you. So as you all know, microglia has many functions, and we are particularly interested in the molecules in the surface of the microglia that coordinates the response of the microglia to different stimuli, sensing, for example, as you saw in the previous presentations, like inflammation, infection, etc., or tissue damage. So we are interested in immune receptors, especially those that are related to clearing the extracellular space of dying neurons, of myelin, cellular debris, or even bacteria, etc., and their relation to regulating the general phenotype of the microglia. And as we know, microglia comes in many flavors, and they coexist even in the same tissue in the same moment, so it's a difficult task. So I've been working in different immune receptors. We are mainly interested now in this. You see my pointer. I hope you see my pointer. We are interested in this family of lipid binding immune receptors. You probably know TREM2, which is very, very well known. There are lipid binding immune receptors that have many different ligands and that confer a specific signaling to the cell that probably coordinates many cell functions. During, as you have seen recently in the other publications, during inflammation, you can have different mechanisms going on, and in particular during chronic low-grade inflammation, like in aging, you will have let's say, a amplifying mechanism that is long-lasting, not very strong probably, but that accumulates with time, and that is in the basis of many age-related diseases like cancer, dementia, etc. So we are interested in this type of processes during aging, and we try to understand how we can modulate, or how microglia modulates these phenotypes with time. So, as you know, this hallmark of aging, this inflammation, has been shown by different ways. But some years ago, a paper came out showing that an inhibitory immune receptor called celiac-E. It recruits phosphatases, and that's why it's an inhibitory immune receptor. And when you do a knockout of this mouse, and then you age this mouse normally, let's say you don't use any aging model, you just wait, for 2-3 years. And then these researchers show by many ways that these animals had a reduced lifespan, but also many characteristics related to aging, like hair graying and many others, increased oxidative stress, etc. So that made us think that these immune receptors could be really involved in normal aging processes, even in healthy aging, and also in neurodegeneration and dementias. So we are focused on this family of immune receptors called CD300. You can see here that they have activating immune receptors, activating members, and also inhibitory members. The activating receptors recruit DAP12, DAP10, similar to TREM2, and the inhibitory receptors recruit phosphatases. So they will dephosphorylate many cascades activated by activating receptors, but also cascades activated, for example, by toll-like receptors. And then we have CD300F here in the corner, which is a bit particular because it has a very big cytoplasmic tail with many different domains that not only recruit phosphatases like SHP1, SHP2, but also GRAB2, for example, Fc receptors and PY3 kinase, etc. So it's a dual receptor that we think that may coordinate many different signals into the microglia. We are interested in the receptor also because it has been shown that it is involved in many inflammatory conditions, mainly outside the CNS, but also in the nervous system, as I will show you partly now. So these lipid-binding immune receptors are very complex. If you see the ligands that they have, the ligands are really not surprising, but really variable. They interact with lipids, so they normally interact with phospholipids, so they can sense phosphatidylserine, for example, and participate in the phagocytosis of dying cells, or decorated beta plaques with phosphatidylserine. They can interact with myelin, interact with sphingomyelin, etc., and with many other lipoparticles that you can find in the parenchyma and in the circulation, etc. And for TREM2 in particular, it has been shown that it can interact with A-beta also and LPS. In the case of CD300F, it's not so clear, but we are trying to understand that also. So they will transduce these signals into the cell, and we'll see now also that they will participate in many metabolic pathways of the cell. So, but which would be the function of these receptors? One of the hypotheses is that these receptors act as dump or pump receptors, and that's why they recognize so different ligands. But also they should have a biological function in the normal CNS. What do they recognize there? Maybe they participate in myelin recycling, the normal myelin recycling, also is synaptic pruning, etc. So we try to understand all these conditions, let's say, and understand how they work. In the first part of the talk, I'm going to show you data related to aging. What we asked was, does immune receptors, in particular CD300F, have an important effect during aging, during normal aging? And what we did was to use knockout mouse and try to understand if the alteration in myeloid cells, including microglia, would affect aging in general in the whole body. So we took knockout mice, we aged them for 30 months, so there's very long experiments, and then we follow them very closely. So when we follow these mice, the first thing that we could see with time was that they have a reduced lifespan. This happened in males and females. It happened in different animal facilities, like in the Universidad de la República, at the Faculty of Medicine, but also at the Institut Pasteur. We generated a new CD300F knockout mouse line, and they also had a reduced lifespan. So we were pretty comfortable that this is really happening and that is a real biological phenomenon, let's say. And then we tried to understand why these animals has this reduced lifespan and health span. So we analyzed many parameters that people analyze for aging. One of the main causes of death of animals with aging is our neoplasm, and we could see that they have similar level of neoplasm, Probably the C300F knockout mice had this neoplasm coming earlier, but in general, they have similar macroscopic number of neoplasms. We could also look at spleen weight because these mice could develop autoimmunity. And we see that some mice could have an increased spleen weight, but that's very variable. And in general, with aging, you see very variable data because animals has a different biological age, really, a different biological age compared to the chronological age. So they don't age at a similar pace, and that makes the data to be really separate. And then we look at many other variables, for example, hepatic markers, and we didn't see much change there also. What we could see in the animals that we had to sacrifice because of ethical criteria is that they had increased GGT. This enzyme is associated to all-cause mortality, and these mice had a significant increase in this circulating enzyme. And we could see also some metabolic parameters that I will not talk much about, but we could discuss if you want. So we analyzed many parameters related to renal biology, liver biology. Obviously, we could not analyze all the possible causes of death, but our initial conclusion is that these animals are really dying because of multimorbidity causes, tumors, ascites, etc. And that is what we are looking at is normal aging, let's say. Obviously, we cannot discard that there's a specific cause of death that we have not seen. So the next question was whether if this is normal aging, we should see increase inflammation. We can see some increased cytokines in plasma and chemokines, also in the lung, in the liver. We can see also increased infiltrates in the liver with time. We can see increase of infiltrates of macrophages. So in general terms, we could see inflammation in different organs. And then we went to see if that also happened in the brain. These are metabolic data that we are not going to talk about now, but if you want to discuss them, we can discuss them after. So we asked if these animals were aging, they should have cognitive decline. So we did several behavioral tests, neurological tests, let's say, and this is the novel object recognition. And you can see here that mice at 18 months old, they discriminate quite well between the familiar and a new object. But then at 24 months old, the knockout mice cannot recognize a new object, and the wild-type mice can do that, but not as well as at 18 months. So our knockout mice are worse. You can see this in females and males. We did also the Barnes maze for memory. It's a typical test that mice has to, let's say, find the place to hide in this platform. You can see that normally with age, they learn similarly, but then the memory for the test, let's say, for memory to find the place to hide, with time, you can see that the knockout mice need more time to find this place to hide. So they have increased memory problems. So in general terms, we can say that these mice also have some cognitive decline at the level of memory. So what happens in the brain? To try to have a rapid and let's say unbiased look at what was going on in the brain, we did whole brain RNA-seq. This is the whole brain chopped, and we did RNA-seq of the whole brain. We could see many mediators that were increased in the knockout mice, like CCL3 and other inflammatory mediators, even GFAP. So the effect was not evidently not only microglia. And many genes also related to aging, like Cloto and other proteins related to aging. So we could conclude that there was also an inflammation going in the brain, and so this was a general phenomenon. To try to understand better what was going on in this brain, what they did was to do gene set enrichment analysis of the RNA-seq data, and we began looking at disease-associated microglia, DAM, and DIM macrophages, which is other type of macrophages related to aging. And you can see here in the gene set enrichment analysis that we have increased dam phenotype here in dark green, and in the other green, we can see these dim macrophages that are also enriched in this brain. So they have increased disease-associated microglia, increased macrophages recruited to the brain. So that was coincident with what we expected, let's say, for an aging brain. And also, we used another set of genes related to senescence to try to understand if this brain has increased senescence. And you can see that this Sen-Mayo profile here is the senescent profile, and these mice tend to have a increased senescence, at least at the transcriptomic level. Another important characteristic of aging is frailty. Frailty is this condition that has, let's say, an unexplained heterogeneity. Is an accumulation of different problems that you can measure different things. One of them is ataxia, coordination problems that arise normally with aging. And we made determinations of ataxia with wild type and knockout mice, and we can see that they have increased ataxia at 2 years of age. And also we used the RNA-seq of the brains to analyze a gene set enrichment analysis for frailty. And again, these brains are increased for frailty-related genes. So we think that these mice also have an increased frailty. Okay, you also know that in the aging brain, there's accumulation of many proteins and lipids. Among them is lipofuxin, for example, and also lipid droplets inside microglia. So we went to look at lipid droplets inside microglia. As you can see here, the knockout microglia have increased lipid droplet content. This receptor is very interesting at the level of the metabolism of lipids. For example, in work done by Andrés Cowen and Natalia Lago, they show that knockout macrophages incubated with myelin accumulates with time more lipid droplets inside the macrophages, so they tend to phagocytose, but they are promised to degrade all these lipids that they are phagocytosing. And you can see the same in vivo when they do a spinal cord injury, that this spinal cord at the long term has increased the accumulation of lipids in this spinal cord. So this also happens in vivo. And we are now analyzing the levels of lipofuxin with Andrés Camadi and Leonel Malacrida there at the Institut Pasteur to try to understand if microglia is also contributing to the accumulation of this lipofuxin that is not being degraded in the brain. But I don't have data related to that. Okay, so this takes us to one of our hypotheses, that was that this microglia has really important metabolic alterations. And a very nice paper came out some years ago showing that really the metabolism of myeloid cells can induce problems in the metabolism of myeloid cells, can induce cognitive decline in aging mice. This is always in normal aging mice. They are not aging models. They are long experiments. And they show there that both microglia, but also circulating macrophages can induce cognitive decline when they have different alterations in the metabolic pathways. So we had also analyzed that in our microglia and the knockout mice. In this case, we did RNA-seq of microglia isolated from mice that had been injected intraperitoneally with LPS, and then we isolated the microglia and did our bulk RNA-seq of this isolated microglia. All the genes that you can see here in this cartoon are downregulated in the knockout mice compared to wild-type microglia exposed to LPS in vivo. You can see that all the many enzymes of the beta oxidation, but also transporters of fatty acids and lipoproteins. Even C-terminal can bind to lipoproteins. So all these pathways are downregulated. Also, many important enzymes of the glucolytic pathways are downregulated, and many shuttle systems of the mitochondria are also downregulated. So this suggested that these cells had problems to cope with the metabolic status, let's say. And then we did seahorse with microglia isolated from the brain, but also with bone marrow that are macrophages. And we can see that mainly in females, we have always an effect that is stronger in females than in males. You can see here, for example, if you see the extracellular acidification rate, like a proxy of glycolysis, you can see that the knockout microglia has a lower basal glycolysis, also a lower basal respiration, and that there are some other changes also in other parameters of the mitochondria. But in general terms, they have, let's say, important mitochondrial and glycolytic alterations, metabolic alterations in general, and also processing of lipids. So from this paper, we concluded that the deficiency of CD300F in general, in a whole knockout, would alter the populations of many myeloid cells, including macrophages and microglia, that would have problems in the metabolic capacity, and that would stimulate this chronic low-grade inflammatory profile that finally would impact on decreased lifespan and also health span. So then again, we need to understand which are the functions of these receptors really, and how they regulate internally the biology of the microglial cells in a more fine way. So what we did is, if the hypothesis is that they act as disease-associated molecular patterns, if they will sense tissue damage, then if we do a laser lesion to the brain and we follow that by 2-photon microscopy, these experiments were performed by Luciana Negro, in fact, we should see a different profile. So what we did was to do these laser lesions. You can see the microglia reacting, the typical reaction that's been described many years ago, and they project towards the lesion their projections. And if you do the same with the knockout mice, you see that they tend to do the same, but it takes more time and the final reaction is not so strong, let's say. We quantify this here. We analyze also the basal motility of the microglia in the neural parenchyma, and that was not changed. So what is really changed is the reaction towards the lesion. So it's not that they don't have energy for projecting, it's somehow they doesn't sense really well what's going on, probably. We try to do a similar approach, doing a small traumatic injury. This is an injury that really spares the meninges, so it's a very subtle lesion. And we looked for the capacity of the knockout mice to sense apoptotic cells and phagocytose apoptotic cells. Here you can see how the microglia change with time, like they acquire like a more phagocytic morphology in the normal mice. And in the knockout mice, these microglia tend to react much less. They have difficulties to really acquire this phagocytic morphology. We analyze different parameters, but you can see here, for example, in vivo, the percentage of phagocytotic microglia at 2.5 hours after the lesion, and we have decreased number of phagocytotic microglia. We see increased number of dying cells in the parenchyma that are not phagocytosed. We also see, for example, an increased distance between a dying cell and a microglial process in the knockout mice. So it looks like they really don't sense that there is an apoptotic cell that needs to be phagocytosed. And interestingly, also what we see is that once they phagocytose a cell, then they keep the cell, the apoptotic cell inside for a longer time period. So they accumulate apoptotic cells inside, and they have problems to degrade these apoptotic cells. And this probably also impacts on the further phagocytosis of additional cells, as Guillermo told you, that Francesca Peri has shown that normally you phagocytose one by one, and then the cell really uses the gastrosome to degrade all that, and they phagocytose again, etc. And these ones are really accumulating cells, and they have problems to really cope with that. So in this sense, we think that they also have problems to really detect apoptotic cells, and after phagocytosing them, also to process them. Okay, I don't have time to show you, but we have seen similar things, that these receptors also participate in the pruning of synapses. but we won't go into that now. So looking at the effect of this receptor in aging and the pathways in which it's involved, it very rapidly came out the idea that it could be they could also participate in Alzheimer's disease, for example, no? Because if you see many of the genes related to Alzheimer's disease, like risk genes, they are part of the endophagocytic pathway. And if you see, for example, the ligands that are putative ligands for these immune receptors, like TREM2, for example, you see there's the same ligands as for C300F. So C300F could really be part of this mechanism and be there as another player that could contribute probably to protect against Alzheimer's disease. So we began to follow that path. We had also other indications that C300F could be part of a similar mechanism. For example, you can see here the TREM2, as you know, is one of the main risk genes for Alzheimer's disease. It's also a lipid-binding immune receptor, similar to C300F. And for example, if you look at the ligands, there are lipids, phosphatidylserine, lipoproteins, so they share similar ligands. If you look at the intracellular signaling pathways, C300F has an ITIM motif, like that recruits SHP1, SHP2. It activates PY3 kinase, also phospholipase C. So, and it also has, C300F can also be cleaved by gamma secretase and release soluble C300F. So there are many shared mechanisms that suggest that it could be part of similar signaling pathways and phenomena. So our hypothesis was that if we now again use knockout mice, we could try to understand which would be the role of this immune receptor in AD. So the first thing we did was we crossed these mice with the 5X FAD mice. This is a model of amyloidosis of Alzheimer's disease related to APP. And normally with these mice, what you do is, if you see the wild-type mice, and this is the elevated plasmase, you follow the time they are in the open arms or closed arms. Normally, these mice increase the time they spend in the open arms. You can see here the contour of the wild-type and the 5X FAD mice, males and females. And as you can see, when we cross them with heterozygosis or homozygosis, they tend to show increased time in the open arms, and that in this model is a higher impact, let's say. We are now waiting, these mice are arriving to 9 months old, so we are measuring now memory. Andres is now doing these experiments, Andres Cowen, and we're looking for novel object recognition, Barnes maze, etc., to see if we have an effect on memory. But it looks like they are worse, let's say. We also know, for example, that in culture, if we culture bone marrow, there are macrophages, and we expose them to A-beta, that they will phagocytose less A-beta. And then there were a couple of papers that were very interesting relating C300F with Alzheimer's disease, at least in animal models. There's the group of Michael Heneka, and they showed both in a tau mice model, but also in a A-beta mouse model, that if you knock out the inflammasome, the NLRP3 inflammasome, this is neuroprotective against tauopathies or beta amyloid pathologies. And when they go and look for the genes that are expressed in microglia that could be related to this neuroprotection in the knockout mice of inflammasome, the very few genes come out, and you see that one of them is C300F, that is induced in the NLRP3 knockout mice, and that suggests that it could be a neuroprotective genes related to this neuroprotection observed when you knock down the inflammasome. And if you see the expression of different genes in this model, you can see that C300F always pops up there like an interesting gene. Again, they did the same in the A-beta mice. And again, when they look for the genes that could be more relevant, crossing different data of single cell and bulk RNA-seq, they end up with very few genes that could be the core genes related to the neuroprotection in the knockout of the inflammasome. And you see again, C300F appears here upregulated in the knockout. So this suggested that that this could be an interesting gene to take into account in Alzheimer's disease. And again, also in this paper, this is a very interesting paper because they show that the inflammasome could also have effects regulating the metabolism of the cell independently on the inflammatory reaction, let's say. They show, for example, that knockout cells, you see THP1 cells is a model of human monocytes, it's a cell line, that they have decreased metabolic capacity, let's say, in general, when they don't have the inflammasome. And they also follow which path could be related to the effect of this inflammasome at the level of metabolism. They show that the inflammasome could be associated with mitochondria. And what it really does is modulating this gene. This is, in fact, this is a glutamate transporter, the typical GLAST AT1 astrocytic glutamate transporter that the microglia also have, and it could be also part of the mitochondrial membrane. So it will shuffle the glutamate into the mitochondria, shuffling in the alpha-acetoglutarate to the Krebs cycle, fueling, let's say, the mitochondria in that way. But also, they also show that it could also have some effect at the level of epigenomics. But again, what they show is that the NLRP3 could really be involved in regulating the metabolism of the cell and in a flux that would include C-terminal-F and this glutamate transporter and alpha-acetoglutarate to get into the Krebs cycle. So we follow this experiment. For example, you can see here when we use, again, bone marrow, there are macrophages, and we treat them with, try to activate the inflammasome. We treat them with LPS here. And you can see that the inflammasome, the expression of NLRP3 is much more increased in the knockout mice compared to the wild-type mice. So the knockout mice have increased activation of the NLRP3, or at least transcriptomics of the NLRP3. If we go and analyze the production of IL-1 beta, you can see here that the knockout mice produce more IL-1 beta in response to LPS plus ATP for releasing IL-1 beta. And the same happens up to a lower extent because this is A beta, not ATP, but you can see that the knockout mice also release more IL-1 beta than the wild-type bone marrow macrophages. So again, when you knock down CD300F, you will see that the inflammasome in general tends to be more active. And also, when we look at the RNA-seq data, we also see that the glutamate transporter, the GLAST1, is also decreased in our knockout microglia. So maybe a similar mechanism as described here in these mice for AD could also be happening in the knockouts for CD300F in the same way. So we try to understand also if this happens in humans. So one of the ways is to use iPSCs in collaboration with Rubén López Vales and Joana Garcia. Andrés Cowen did these experiments. They differentiate iPSCs to microglia, and then we treat them with a contra antibody or an antibody that activates CD300F. In this way, we trigger CD300F, and we also treat them with this microglia with A beta or A beta plus LPS. You can see here that A beta plus LPS induce the production of IL-1 beta, and when you trigger CD300F with activating antibodies, you can reduce this production of IL-1 beta. So it's really strongly pro-inflammatory. Okay, so trying to understand if this works in humans, we also work with THP1 cells. We are analyzing transcriptomics in these cells. We see that, for example, when we activate CD300F, there's a regulation of different genes related with the Alzheimer's disease risk genes. So it's an enrichment in Alzheimer's disease genes. And then we took advantage of a SNP. In fact, it's an allele that is quite prevalent in the general population. We know that an allele of CD300F that changes an amino acid in the cytoplasmic tail can change the signaling. of the receptor. And we have shown that this changes, this kind of effect in humans, for example, in depression in females and in anxiety in males. So we went further to try to test this in Alzheimer's disease. So we went to the ADNI database. This is an open source database which has a lot of data related to Alzheimer's disease patients. And we screen for this allele. What you can see here is that there's a significant association of this allele with a better neuroscore of Alzheimer's disease. This is the CDR sum of boxes score. And if the patient that has this allele has a less evolved Alzheimer's disease, let's say. So this was very encouraging. We controlled this for sex, ethnicity, age, etc. And the effect was still there. You can see this effect even in heterozygosis. So it's some gain of function of the receptor. And this is also interesting when you look at biomarkers for Alzheimer's disease. You can see that in the patient that has this allele, you have decreased tau in the CSF, which is good. You have increased A-beta in the CSF, which is also good, especially when you stratify the patients by sex. You see that mainly in men and not in females. And also you have an increase in the fluorodeoxyglucose in the PET in the hippocampus, which is also good. So this SNP this allele is associated to better biomarker, let's say, profile. Then we did the same with another validation cohort at the Hospital Clinic in Barcelona. We genotyped these patients and we collaborated with all the group of Ana Antonella, Raquel Sanchez Valle to see this. And again, we see similar results. So again, the patients that have this allele are more protected, let's say, against the progression of the disease. So now what we have done humanized mice that overexpress the human CD3F or this allele, the R218Q is this allele, and we're going to cross them with tau mice model and A-beta mouse model in collaboration with Michael Heneka and do these experiments to try to see these humanized mice if we see similar results. Don't worry about that. Just as a last slide to not take more time about this talk. We are doing immunoprecipitation to try to understand which protein are associated to the cytoplasmic tail of this receptor that has this substitution. So what we do is we express the normal human receptor or the allele, and then we immunoprecipitate in mild condition, let's say, to try to understand which protein associates with the receptor in the cytoplasmic tail. And then we do proteomics and see which proteins are there. For example, we see more GRAB2, which is one of the proteins that should be interacting with the CD3F. We see annexins that are always there, that are probably related to membrane dynamics. And also we see some interesting glycolytic and enzymes that suggest that maybe the receptor is recruiting locally some energy producing hubs, let's say, that could be generating local energy there. But this is just a hypothesis. So as always, the most important slide, this was our group in Uruguay, and most of this, all the aging was done in Uruguay with this group, mainly by Frances Evans, that I think is there in the meeting. But we all worked on that, and this is our group now in Barcelona. Andrés Cowen, you can see him. He's now in Barcelona with us, and finally, this is also all our collaborators and funding agencies. Okay, thank you.
Okay, thank you everyone for the question. Felipe, you have to come here, please. Felipe is going to do a question.
Hola, Hugo. Hi,
Hola.
Felipe. Yeah, I found very interesting the data. You have to, I don't know who want to be Google. Okay, yeah. I found very interesting the data about Alzheimer's, especially the variants. About the aging data, I'm still trying to kind of understand what is the connection between this receptor and aging, because you take it out from the system and the animals die early. Then I'm trying to kind of think that in a normal animal, this receptor goes down normally in aging. Then basically, if you upregulate it, for example, with an overexpression strategy, you will get like a longer lifespan or health span. Yeah, that's kind of my question.
Yeah, normally it goes a bit up during aging, so it's upregulated during aging normally. We would love to do the experiment of overexpressing it and age the animals. What I can tell you is that very recently another paper came out on TREM1. TREM1 is a activating immune receptor instead of inhibitory immune receptor, and in that paper they show that if they knock out TREM1, the animals live longer. So it's quite logical that if you knock down inhibitory immune receptors, the animals live less, and if you knock down activating immune receptors, they live longer. That's the only thing that I can tell you in general. And also that from our point of view, we have looked at different possible pathologies that could be related to the knockdown of the receptor, and we don't find any specific thing. So it looked more like accelerated aging, but it's difficult to say because obviously we cannot rule out every possible effect of the receptor. So.
Okay. Well, thank you very much, Hugo. I think it's late, so we're going to leave here. Is there any more questions related to this? Well, thank you very much, Hugo. Amazing story, and see you soon. Bye-bye.
Great. Thank you. Bye-bye. Have a nice time there.
Three or small things before go to the focus section. First of all, I forgot this morning something very, very, very important, and I'm really very grateful. I don't say thank you to the organizing committee integrated by Dr. Patricia Zeta. Please stand up. I know that you go ahead. Don't be the committee was integrated by Dr. Correales and young people take care of us, and they work and we do all the things in them because they work very, very hard, and I do almost nothing. Thank you very much for that. The other thing that I forgot is to say thank you, Nicolas. Nicolas belongs to the Informatics Center of the School of Pharmacy and Biochemistry, and I guess it's the best in the University of Buenos Aires. I know that it's, I don't know, probably he's not happy because he's here from the 8 o'clock in the morning until now. But anyway, he's a wonderful person, a wonderful professional, and for that, I ask a clap for him. Okay? I have somebody told me, why don't you write somebody to publish abstract of his meeting? And I had the idea of send an email to Dr. Lawrence, Andres Lawrence, that is the chief editor of the Journal of Neurochemistry. Immediately, Andy answered me and said, of course, I published the abstract of the meeting, but I have another idea. I want to help and help the Club of GLIA, the Latin American Club of GLIA, and I decided to prepare a special issue on this, in this workshop that the Club and Latin American GLIA Club is organizing. For that, the special issue was integrated by the abstract. Then the short story of the Latin American GLIA Club. Then all the lectures, all the invited lectures receive an invitation for writing a short version of the specification that you have here. Please, I ask you, help the GLIA Club and write this version, and they ask you. Then every year receive another mail asking for write on the cell and invite the symposium speaker for write also a short story of that presented in this symposium. Also, I ask that do it. The deadline is August. You have time to do it. And finally, we put there the story of the neurochemistry in Latin America. I have written this story, but anyway, I said to Patricia, to Flavia, and to somebody in Chile for corrected and adding or doing something for doing a whole story. And as I never am quiet, I decided that I want to integrate Mexico in this Latin America GLIA Club. For that, I wrote to a friend of mine, Dr. Arturo Ortega Soto, and he said, of course, I will write the story of the neurochemistry. I hope that Trump don't decide to colonize Mexico. So that's it, because the GLIA Club is named now Latin American GLIA Club. He said, he promised, I don't know, that he's trying to get money for consolidate the integration of Mexico in the GLIA Club, doing something in Mexico similar to this. I hope that he got the money, and we have in the room the next treasurer of the International Society of Neurochemistry, Dr. Vlad Porpura. Go ahead, put a here. He is a Dr. Ortega was selected a member of an officer of the society. So we hope that we can do it. Please write all that we ask, because it's very important for us, this special issue in strong support of this in Latin American GLIA Club. So you have a little bit of caffeine for support the poster discussion, and go ahead immediately.
Yes.
It's very bizarre. Just make it fun. And it works beautiful. And when I start to get here, I passed this school with the secret. I don't know why it started to hang out with this is cool for oh yes because you selected yeah no but I do like that they take smaller little shots but they once you start to do this for
Me da vergüenza, pero me paro porque si no, no terminamos. Lo que pasa es que ellos, a ver,
¿qué querían? ¿El puntero?
Claro. No sirve para acá sí. Nosotros somos científicos, pero nosotros tecnológicos, con lo cual.
Sí, yo soy tecnológico en lo que yo sé.
Claro,
Cosas que
ellos nosotros. Yo a lo mejor. Pero lo dejo hacerlo porque la gente
lo otro sí, ¿sabe inglés?
Ah, bueno, pero yo lo conozco, que lo meta.
Que lo meta y que te corran los momentos.
Oíme una cosa. Todas estas cosas, todas estas
Yo esto me lo sigo.
entonces se llevan
No, pero ahorita les digo, Darío supuestamente se fue.
Dejemos entonces también esta computadora junto con todo eso bajo llave, así yo mañana ya vengo directo acá, porque si no tengo que ir a
Pero para
llevarla.
vos que decís acá,
No, donde vos dejes la tuya.
yo voy a dejar todo arriba en el C5.
Deja esta también, por favor. Porque esta es la del departamento.
En realidad yo precisaría separar lo que es transmisión de presentación. Porque para mí que no sé qué, algo tocaron, que ahí me hicieron la regla.
Puede ser.
Porque no viste que al principio salieron, las dos
Claro,
primeras salieron, los
sí.
tres, todos salieron bien, o sea, faltaron
Yo no
cositas.
sé si era él también que, yo no sé, porque no tocamos nada entre él y los anteriores.
No, por eso. Yo no sé si
Es
era
algo
algo.
de él. A mí me parece
No,
que era
no sé,
algo
la
de
verdad.
él.
Si te digo la verdad, te miento.
Yo creo que era algo de él porque nosotros no tocamos nada de... Después de él, de los otros dos no sacamos nada.